modified cephalosporin antibiotic molecules and diagnostic radiopharmaceuticals based on these molecules for imaging bacterial infections, their preparation and use. Przemysław Koźmiński, Ewa Gniazdowska, Paweł Halik, Kinga Żelechowska-Matysiak, patent PL number 242808. Radiopharmaceuticals are biologically active compounds labeled with radionuclides. Modern advanced radiopharmaceuticals are used in targeted therapy, which means that they are designed to find the molecular target in a patient's body and accumulating it. Depending on the used radionuclide, we can divide radiopharmaceuticals into two groups, diagnostics and therapeutics. For therapy, emitters of alpha, beta, and Auger electrons are used. These radiations are characterized by short range and high linear energy transfer, enough to cause double DNA break, generate free radicals, or destroy cell membranes. For diagnosis, we use emitters of penetrating gamma radiation that can be emitted directly or indirectly in the annihilation of positrons. The place of accumulation of diagnostic radiopharmaceuticals is determined by external gamma cameras. The subject of the invention is the use of diagnostic radiopharmaceutical for imaging infections caused by G+ and G- bacteria, especially for imaging and diagnosing infections related to diabetic foot syndrome. Scintigraphic studies using a labeled antibiotic from the cephalosporin group have demonstrated higher sensitivity and specificity compared to standard methods. In order to modify the antibiotic linker, example aspartic acid ASP with a blocked amino group is converted into anhydride form. Then the prepared compound is mixed with an antibiotic from the cephalosporin group in DMF solvent. After a minimum of 12 hours of synthesis at room temperature, the linker is attached to the antibiotic. After synthesis, the compound is treated with a piperidine solution to remove the amino-protecting group. The modified antibiotic molecule from the cephalosporin group is combined with a selected macrocyclic or linear ligand. DOTA, DOTAGA, NOTA, NODAGA, TRAP, NOPO, TCMC, DTPA, THP, HBD, DFO, or its derivative. This compound forms complexes with transition metal cations, 94M technetium, 99M technetium, 68 gallium, 111 indium, 89 zirconium, 43 scandium, 44 scandium by attaching the ligand to the antibiotic molecule through an amide or thiourea bond between the carboxylic or isothiocyanate group of the selected ligand and the primary amino groups. The use of the linker and the diagnostic radionuclide labeling does not alter the biological properties of the antibiotic. Cephalosporins constitute a group of semi-synthetic beta-lactam antibiotics with a broad spectrum of bactericidal activity against both gram-positive and gram-negative bacteria, acting by inhibiting bacterial cells wall synthesis. The radiopharmaceutical comprising the modified antibiotic molecule from this group is intended for imaging infections caused by gram-positive and gram-negative bacteria, especially for imaging and diagnosing infections associated with diabetic foot syndrome. X-ray imaging does not show inflammatory changes in bones during the first 2 weeks of infection, and magnetic resonance imaging may not differentiate between infection and aseptic inflammation. Scintigraphic studies using a labeled antibiotic from the cephalosporin group exhibit higher sensitivity and specificity than standard methods. The application of the antibiotic labeled with a diagnostic radionuclide innovatively leverages the known action of the antibiotic and does not require significantly high cost, thus enabling wide accessibility to this type of diagnostic.